In the previous parts of this GPCR series, we explored the structure of G protein-coupled receptors, their activation mechanisms, the different G proteins, second messenger signaling pathways, the major GPCR receptor families, and their clinical and pharmacological importance. By now, you know that receptors such as the adrenergic, muscarinic, dopamine, histamine, serotonin, opioid, and angiotensin receptors all belong to the GPCR superfamily. This naturally leads us to an important question. If all of these receptors belong to the same GPCR superfamily and share the same basic seven-transmembrane structure, why are they divided into different classes? That is exactly what we will discuss in this video. Unlike our previous videos, this lecture is not about GPCR signaling. Instead, it focuses on how the GPCR superfamily is organized and why scientists classify these receptors into different groups. Understanding this classification helps us appreciate how receptors with the same basic architecture have evolved to recognize different ligands and regulate a remarkable variety of physiological functions. Before discussing the individual classes, let us first understand why GPCR classification is necessary. Although all GPCRs possess the characteristic seven-transmembrane, or 7-TM, architecture and transmit signals by activating intracellular G proteins, they are not identical. Over millions of years of evolution, GPCRs have diversified considerably. Some receptors recognize small neurotransmitters, while others bind large peptide hormones, amino acids, calcium ions, developmental proteins, odorants, or even light. Because these receptors recognize such diverse ligands, their structures have also evolved in different ways. Some have very small extracellular regions, whereas others possess large extracellular ligand-binding domains. Some function as single receptor molecules, while others must form dimers to become active. Therefore, grouping all GPCRs into a single category would overlook these important structural and functional differences. To make their organization easier to understand, GPCRs are classified into different groups based on the characteristics they share. So, on what basis are GPCRs classified? The classification is based primarily on four major characteristics. The first criterion is structural organization. Although every GPCR contains 7 transmembrane helices, the arrangement of their extracellular domains, intracellular regions, and ligand-binding sites differs among receptor families. These structural differences influence how receptors recognize ligands and initiate signaling. The second criterion is the amino acid sequence. Receptors with similar amino acid sequences usually share similar structural features and often evolved from a common ancestral receptor. Comparing amino acid sequences allows scientists to group closely related receptors into the same family. The third criterion is evolutionary relationship. GPCR classes are not arbitrary groups. Instead, they represent receptor families that have evolved from common ancestral genes over millions of years. As evolution produced new receptors, they retained certain structural features while adapting to recognize different signaling molecules. This explains why receptors within the same class resemble one another more closely than receptors belonging to different classes. The fourth criterion is the mechanism of ligand recognition. One of the most important differences among GPCR classes is where and how the ligand binds. For example, some receptors bind ligands within the transmembrane helices. Others bind ligands through a large extracellular domain. Some require two receptor molecules to work together. Others recognize specialized developmental signaling proteins through unique extracellular domains. These differences in ligand recognition are among the defining characteristics of each GPCR class. Before moving further, remember one very important concept: GPCR classification is not based on the type of G protein that a receptor activates. This is a common misconception. For example, receptors belonging to Class A may couple to GS, GI, GQ, or G12-13, depending on the receptor subtype. Similarly, receptors belonging to the other classes may also interact with different intracellular signaling proteins. Therefore, GPCR class does not determine the G protein. In modern receptor biology, GPCRs are commonly classified using the GRAPHS classification system. The name GRAPHS is an acronym representing the 5 major GPCR families. G stands for glutamate, R stands for rhodopsin, A stands for adhesion, F stands for frizzled, and S stands for secretin. In many pharmacology and physiology textbooks, however, the adhesion GPCRs are discussed together with the secretin family because they share important structural similarities. As a result, many introductory courses describe 4 major GPCR classes: Class A, rhodopsin-like receptors; Class B, secretin family, including adhesion GPCRs; Class C, Metabotropic glutamate family; and Class F, Frizzled family. This is the classification that we will follow throughout this lecture. Now that we understand why GPCRs are classified and the principles behind this classification, we can begin exploring each class in detail. We will start with Class A, also known as the rhodopsin-like family, because it is the largest, most diverse, and most clinically important GPCR class. As we study each class, instead of simply memorizing receptor names, focus on 3 key questions. What makes this class structurally unique? How does it recognize and bind its ligands? What are its major physiological and clinical roles? Keeping these questions in mind will make it much easier to understand the similarities and differences among the GPCR classes. Class A is the largest GPCR family, accounting for approximately 80% of all GPCRs. It also contains the majority of receptors encountered in medicine and pharmacology. Because of its enormous diversity, Class A regulates almost every major physiological system in the human body. To understand why this family is so successful, let us first examine its structural features. Although Class A receptors share the common 7-transmembrane architecture found in all GPCRs, they possess several structural features that distinguish them from the other GPCR classes. One important characteristic is their relatively short extracellular N-terminal region. Unlike some other GPCR classes, Class A receptors generally do not require a large extracellular domain to recognize their ligands. Instead, the ligand-binding site is usually located within the transmembrane region, forming a pocket created by the 7-transmembrane helices. Another important feature is the presence of several highly conserved amino acid motifs. These conserved sequences have been preserved throughout evolution because they play essential roles in maintaining receptor stability and facilitating receptor activation. 3 of the best-known conserved motifs are DRY motif, located near the cytoplasmic end of the 3rd transmembrane helix, consists of aspartate, arginine, and tyrosine. In the inactive receptor, it forms an ionic lock that stabilizes the receptor. Upon ligand binding, this lock breaks, allowing G protein binding and initiating GPCR signaling. CWXP motif, located within the 6th transmembrane helix, consists of cysteine, tryptophan, any amino acid, and proline. It functions as a molecular toggle switch, causing TM6 to move outward after ligand binding. This creates the intracellular pocket required for G protein coupling. NPXXY motif, located near the intracellular end of the 7th transmembrane helix, consists of asparagine, proline, 2 variable amino acids, and tyrosine. It acts as a conformational anchor, stabilizing the receptor in its active state and supporting efficient G protein signaling. Rather than memorizing their exact locations, remember their functions. Together, these conserved motifs help maintain the receptor in its inactive state, stabilize the active conformation after ligand binding, and promote efficient interaction with intracellular G proteins. Now that we understand the structural organization of Class A receptors, the next question is, how do these receptors recognize such a wide variety of ligands? One of the defining characteristics of Class A receptors is where the ligand binds. For most Class A receptors, the ligand enters a binding pocket formed by the seven-transmembrane helices. This is quite different from some other GPCR classes, where ligand binding occurs mainly in large extracellular domains. When the ligand binds within this transmembrane pocket, it induces subtle conformational changes in the receptor. These structural changes are transmitted through the transmembrane helices to the intracellular surface of the receptor. As a result, the receptor becomes capable of interacting with a heterotrimeric G protein, initiating intracellular signaling. Since we have already discussed the detailed activation mechanism and G protein signaling pathways in the previous videos, we will not revisit those mechanisms here. Instead, the key point to remember is that the transmembrane ligand-binding pocket is the structural hallmark of Class A receptors. This unique design enables these receptors to recognize an extraordinary variety of signaling molecules. So, what kinds of ligands can Class A receptors recognize? Class A receptors interact with the widest variety of ligands among all GPCR classes. These include neurotransmitters, such as adrenaline, noradrenaline, acetylcholine, dopamine, serotonin, and histamine. Hormones, including angiotensin II peptides, such as endogenous opioid peptides. Lipids, including lysophospholipids and other lipid mediators. Chemokines, which regulate immune cell migration, odorant molecules detected by olfactory receptors, and light, detected by rhodopsin in the retina. This remarkable diversity of ligands explains why Class A receptors are involved in so many physiological processes. Having understood the types of ligands they recognize, let us now look at the major receptor families that belong to Class A. The majority of GPCRs studied in pharmacology belong to Class A. Some important examples include adrenergic receptors, muscarinic receptors, dopamine receptors, histamine receptors, most serotonin receptors, with the important exception of the 5-HT3 receptor, which is a ligand-gated ion channel, rather than a GPCR, opioid receptors, angiotensin receptors, chemokine receptors, and olfactory receptors, rhodopsin, the visual receptor responsible for dim light vision. You have already encountered many of these receptor families in earlier modules of this receptor series. Now let us see how these receptors contribute to normal physiology. Because Class A receptors respond to such a diverse range of ligands, they regulate almost every major physiological system in the body. Their functions include regulation of heart rate and blood pressure, control of smooth muscle contraction and relaxation, neurotransmission throughout the central and peripheral nervous systems, endocrine and exocrine gland secretion, vision through rhodopsin, olfaction through odorant receptors, immune cell migration through chemokine receptors, pain perception through opioid receptors, and regulation of metabolism and energy balance. This broad range of functions makes Class A receptors indispensable for maintaining normal physiological homeostasis. Naturally, receptors that regulate so many essential processes are also of tremendous clinical importance. Class A receptors represent the largest group of therapeutic targets in modern medicine. In fact, a substantial proportion of currently approved drugs exert their effects by acting on Class A GPCRs. Examples include beta blockers, which target beta-adrenergic receptors to treat hypertension, angina, and heart failure, beta agonists, used to produce bronchodilation in asthma and chronic obstructive pulmonary disease, antihistamines, which block H1 receptors to treat allergic disorders, opioid analgesics, which activate opioid receptors to relieve severe pain, angiotensin receptor blockers, ARBs, which inhibit AT1 receptors in hypertension and cardiovascular disease. Dopamine receptor antagonists, widely used in the management of schizophrenia and other psychiatric disorders. These examples illustrate why Class A receptors dominate both pharmacological research and clinical therapeutics. As we have just seen, Class A receptors recognize most of their ligands within the transmembrane region. However, not all GPCRs follow this strategy. Some receptors bind much larger signaling molecules, particularly peptide hormones. To recognize these larger ligands, they require a different structural design. This brings us to Class B, also known as the secretin family. Although Class B receptors share the same seven-transmembrane architecture as all GPCRs, they differ markedly from Class A in both their structure and ligand-binding mechanism. Let us first examine their structural features. Like all GPCRs, Class B receptors possess 7-transmembrane A helices. However, their most distinctive feature is a large extracellular N-terminal domain, which is considerably larger than that of Class A receptors. This extracellular domain contains several conserved cysteine residues that form disulfide bonds, helping to stabilize the receptor's three-dimensional structure. Unlike Class A receptors, where the ligand-binding pocket lies mainly within the transmembrane bundle, the extracellular domain of Class B receptors serves as the primary site for peptide hormone recognition. This unique structural arrangement allows these receptors to bind large peptide ligands that would not fit into the relatively small transmembrane binding pocket of Class A receptors. Now that we understand their structure, let us see how these receptors recognize and bind their ligands. Class B receptors employ a characteristic two-domain binding mechanism. In the first step, the peptide hormone binds to the large extracellular N-terminal domain. This initial interaction positions the ligand correctly on the receptor. In the second step, part of the peptide interacts with the transmembrane domain, producing conformational changes that activate the receptor. Once activated, the receptor undergoes conformational changes that enable it to interact with intracellular G proteins and initiate signaling. The important point to remember is that ligand recognition begins outside the transmembrane region, unlike Class A receptors, where ligand binding usually occurs within the transmembrane helices. Thus, the large extracellular ligand binding domain is the structural hallmark of Class B receptors. Having understood their activation mechanism, let us now look at the types of ligands recognized by this family. Class B receptors primarily recognize large peptide hormones. These peptide hormones function as extracellular signaling molecules that coordinate communication between different organs and tissues. Examples include hormones involved in glucose regulation, calcium homeostasis, gastrointestinal function, bone metabolism, stress responses, and energy balance. Because peptide hormones are relatively large molecules, they require the specialized extracellular binding domain found in Class B receptors. Let us now examine some representative members of this receptor family. Important examples of Class B receptors include secretin receptor, glucagon receptor, GLP-1 receptor, GIP receptor, calcitonin receptor, parathyroid hormone receptor, corticotropin-releasing hormone receptor, vasoactive intestinal peptide receptor, and pituitary adenylate cyclase-activating peptide receptor. Although these receptors recognize different peptide hormones, they share similar structural characteristics and ligand-binding mechanisms. Now let us examine the physiological roles of this receptor family. Class B receptors are primarily involved in endocrine and metabolic regulation. Their major physiological functions include regulation of blood glucose levels, stimulation of insulin secretion, control of glucagon release, appetite regulation and satiety, gastrointestinal secretion, bone remodeling, calcium homeostasis, stress adaptation, and hormonal regulation. Unlike Class A receptors, which participate in numerous rapid physiological responses, Class B receptors mainly regulate long-term hormonal communication between organs. Because of these essential functions, they have become important therapeutic targets. Several important modern drugs act on Class B GPCRs. Examples include GLP-1 receptor agonists, widely used in the treatment of type 2 diabetes mellitus and obesity. Parathyroid hormone analogs, used in the treatment of osteoporosis. Drugs targeting calcitonin receptors are used in disorders of calcium metabolism. And experimental therapies targeting glucagon receptors for metabolic diseases. In recent years, Class B receptors have become one of the fastest-growing areas of GPCR drug discovery because of their central role in metabolic disorders. The third major GPCR class adopts an entirely different strategy. Rather than functioning as individual receptor molecules, these receptors must pair together to form a functional receptor. This brings us to Class C, also known as the Metabotropic glutamate family. Like all GPCRs, Class C receptors possess 7-transmembrane A helices. However, their extracellular region is much larger than that of Class A receptors and contains a unique ligand-binding domain known as the venous flytrap or VFT domain. This domain derives its name from its resemblance to the leaves of a venous flytrap plant. Another defining characteristic of Class C receptors is that they function as obligate dimers. This means that two receptor molecules must associate to form one functional receptor. A single receptor molecule alone is generally unable to function normally. Dimerization is therefore an essential feature of this GPCR class. Now that we understand their structural organization, let us see how these receptors become activated. Unlike Class A receptors, where ligands bind mainly within the transmembrane region, Class C ligands bind to the venous flytrap domain. When the ligand binds, the two lobes of this extracellular domain close around the ligand, much like a venous flytrap plant closes around its prey. This movement produces conformational changes that are transmitted through the receptor to the transmembrane helices. These structural changes then activate the receptor, allowing it to interact with intracellular signaling proteins. The important concept to remember is that ligand binding, domain closure, dimerization, and receptor activation are closely linked in Class C GPCRs. Compared with Class A and Class B receptors, Class C receptors recognize a more specialized group of ligands. These include amino acids, neurotransmitters, calcium ions, and taste molecules. This reflects their primary roles in nervous system function, mineral homeostasis, and sensory perception. Major members of Class C include Metabotropic glutamate receptors, GABA-B receptors, calcium sensing receptor, sweet taste receptors, and umami taste receptors. Although these receptors recognize different ligands, they all share the characteristic venous flytrap domain and dimeric organization. Class C receptors play essential roles in several physiological processes, including regulation of excitatory and inhibitory neurotransmission, synaptic plasticity, learning and memory, control of neuronal excitability, maintenance of calcium homeostasis, and detection of sweet and umami taste. These receptors are especially important within the central nervous system, where they help regulate neuronal communication. Because of their involvement in nervous system function, Class C receptors are important therapeutic targets. Examples include baclofen, a GABA-B receptor agonist used to treat muscle spasticity; cyanocalcet, a calcium-sensing receptor activator used to treat secondary hyperparathyroidism. Researchers are also investigating drugs targeting metabotropic glutamate receptors for neurological and psychiatric disorders, including anxiety, depression, schizophrenia, chronic pain, and neurodegenerative diseases. The fourth and final major GPCR class is Class F, commonly known as the Frizzled family. Although these receptors share the characteristic 7-transmembrane architecture of all GPCRs, their primary function differs considerably from the previous classes. Rather than regulating classical neurotransmission or endocrine signaling, Class F receptors control developmental signaling pathways that determine how cells grow, differentiate, and organize into tissues. Their distinguishing feature is an extracellular cysteine-rich domain located at the N-terminus. This domain serves as the principal ligand-binding region and is stabilized by multiple disulfide bonds. Unlike the venous flytrap domain of class C or the peptide binding domain of class B, the cysteine-rich domain is specialized for recognizing developmental signaling proteins. The two principal members of this family are frizzled receptors and smoothened. Frizzled receptors bind Wnt proteins, initiating Wnt signaling pathways that regulate cell proliferation, differentiation, migration, and tissue patterning. Smoothened functions in the hedgehog signaling pathway. Under resting conditions, smoothened is inhibited by another membrane protein called patched. When the hedgehog ligand binds to patched, this inhibition is relieved, allowing smoothened to become active and initiate downstream signaling. Unlike the other GPCR classes, many class F receptors signal through both G protein-dependent and G protein-independent pathways, depending on the cellular context. Major members include frizzled receptors, FZD1, FZD10, and smoothened receptor. Although relatively few in number, these receptors regulate some of the most fundamental biological processes in multicellular organisms. Class F receptors regulate embryonic development, organ formation, stem cell maintenance, cell differentiation, cell polarity, tissue homeostasis, wound healing, and tissue regeneration. Because these pathways control normal growth and development, they must be tightly regulated throughout life. Abnormal class F signaling has been associated with developmental disorders, fibrotic diseases, degenerative diseases, and multiple forms of cancer. Consequently, drugs targeting frizzled receptors, smoothened, and the Wnt and hedgehog signaling pathways are active areas of pharmaceutical research, particularly in oncology and regenerative medicine. Thus, although class F GPCRs represent a relatively small receptor family, they play essential roles in embryonic development, tissue homeostasis, and disease. Their involvement in multiple pathological conditions makes them promising therapeutic targets and an important focus of ongoing drug discovery. With this, we have completed our discussion on the major classes of G protein-coupled receptors. Understanding the unique structural and functional characteristics of each GPCR class provides a strong foundation for appreciating their physiological roles and their significance in pharmacology and therapeutic development. If you found this lecture helpful, please like the video, subscribe to the channel, and turn on the notification bell so you don't miss our upcoming lectures on receptor pharmacology. Thank you for watching, and I'll see you in the next video.